All right, so it's a little after 1 here, so why don't we go ahead and get started. So we have a couple people on the Zoom. Great. Welcome everyone to session 2 of the single cell RNA-seq analysis primer. My name is Tyler Shapey. For those of you who weren't here last time, I'm from the Duke Bird Corps. I'm a biostatistician there, and I'll be covering this. We'll be wrapping up session 1 content, and then we'll move on to the session 2 for today. So why don't we go ahead and get started here with the setup. If you haven't signed in already, please do so with that link. I can put it in the Zoom as well. And then if you could go ahead and launch an on-demand session through DCC. We'll be using the CHSI RStudio Workshop app like we did last time. If you want to request 4 CPUs and 32 gigs of memory, and then use just the R25 HIV image for the singularity image. And that'll get you set up there. And I'll give everybody a few minutes to do that. And while you're working on that, I'll just go over a couple of these intro points here. Kind of similar to last time, I prefer that you ask questions as they come up. If there's anything that's not clear, I'm happy to stop. I want this to be sort of like an informal workshop session. The workshop format, we're going to alternate between slides and code. Actually, yeah, 4 and 32. Yeah. So the workshop will be alternating between looking at slides, and I'll be presenting sort of some of the methods, and we'll alternate between that and the R Markdown file. We do have this kind of tricky situation today where we're going to be working with 2 different R Markdown files, the one from last time and the one from this time. And then kind of same disclaimers as last time. I'm only presenting a sample of the tools and methods. There are a lot of things out there that you could be using. This is sort of just one approach. And then I'll be kind of walking through this as a practical guide from like a practitioner perspective, but I'm not going to be like rigorously proving things or actually even rigorously explaining all the details of every tool that we go through. Obviously, you want to dive into sort of the original papers or other resources if you want to learn more in depth. Okay, so once you have your RStudio session pulled up, recall you'll want to open the RStudio project. It should be 2023-2024 HIV workshop. And the first thing you want to do is to pull the latest content from the repo. I just committed some content this morning, so that that'll get you updated. And I realized that last time, if you had saved any changes you made to the Markdown file, you'll actually get an error when you try to pull regarding uncommitted changes. There are, yes, memory is 32. There are instructions for that situation at the GitLab. If you go to that GitLab repo here from this link, the second link here, there should be a link to instructions for how to pull. And those instructions will have that has instructions for how to overcome this challenge. Yeah, and just let me know if you guys have trouble when you pull. If you get an error message, I can try to help out. And then, yeah, once you are able to pull, if you want to navigate in session 1 to the notebook called the session 1 notebook, and we'll be starting at code section 3, filter cells and genes. And let me actually start the session. Okay, what does it say? Uncommitted. What does the error say? Something about uncommitted changes? Yeah. Yeah, so for that, if you go to this link here. If you go to the second, to the main page for the repo, down here, there is update workshop content. And I think stashing is the best way to do this. So basically what it's saying is if you have changes that you made to any of the files that you're trying to pull, you can't pull the new ones because you have changes that you made that are uncommitted. So if you, actually I can just do this. If you go to, within RStudio Tools terminal, new terminal, should bring up a bash terminal, and then you can do git stash. And I don't have anything to save because I don't have any updates, but if you do that git stash, it should essentially like kind of stash away those changes that you made for later and revert you back to the unchanged version. And then that unchanged version will be able to have the changes pulled into it. Hopefully that works. And you can, I think, recover those changes later if you want to go back. Yeah, okay, great. Okay. And I don't see any comments on the Zoom, so I'll take it to mean that the Zoom folks don't have any problems. Okay, so if we let's go to the session 1 notebook. This, again, will have been updated from last time. What I did was actually put the exercise answer code into each of the exercise chunks, so that what we can do is just go to code section 3, filter cells and genes. And if you just want to scroll down to this chunk here on line 759, and if you hit this arrow here in that chunk, the down arrow, it'll run all of the chunks above. And that'll basically, right, it'll do everything we did last time during the workshop. And it should take it a minute to run, but that'll get us all ready to go for the session. Okay, so is everybody either running all those chunks from the previous chunks or have them run at this point? Yeah. Okay. So we can go ahead and finish up. Actually, I guess I have to wait for it. Oh, I do have a reminder to take a break at the height of the clips, so we can take 5 minutes and go out and take a look at it if we all want to. Okay, Elizabeth has an error at source config assay. What does the error say, Elizabeth? Surprised that that's a spot where you'd have a problem. Okay. Are you, Elizabeth, are you in the project? Like, did you go to this upper right menu and select the project? That could be one possibility. I'm trying to think of what else it could be. Essentially saying that you don't have the code pulled to the kind of correct location. Okay. All right. So Elizabeth says she's going to be right back. I think hopefully she can resolve that. I think I kind of, just for the interest of time, want to get started here. So if everybody else has kind of this quick run, the kind of sections above code section 3, we can resume. I did cover this grep string searching last time. We'll want to run this chunk here on line 774 that calculates the percentage of UMIs mapping to mitochondrial genes, and 795 calculating the percent of UMIs mapping to ribosomal genes. And then we're going to run this chunk that calculates those additional quality control metrics, the log 10 of counts, log 10 number of features, and the percentage of counts in the top 20 highly expressed features. Okay, great. Elizabeth says she's fixed. Perfect. All right, so we can take a look. We looked at these plots last time. These are the violin plots showing kind of the distribution essentially among all the cells, across the cells, for the unfiltered set of cells that we have across the 4 metrics. And then I mentioned this last time, we're going to basically filter on these threshold values based on the median absolute deviations from the median. And okay, so I can walk through this next code chunk. So I guess I'll start here. So again, the strategy is to basically filter out cells that appear to be outliers in at least 2 of these metrics. And we're following sort of the approach that Germain et al. 2021 published. And essentially what we do is we are creating these cutoff values in terms of the median absolute deviations, or MADs, from the median value for each metric. So Germain et al. Kind of provide these threshold scores here based on the MADs. And I'll just go through one of them. So for example, for log 10 UMI counts, we would consider as an outlier a cell with a log 10 counts of less than, a value with log 10 counts less than 5 median absolute deviations from the median. So in other words, lower than the median. And it's basically similar logic for all of the remaining metrics. Okay, so then there's this chunk here where a lot happens. And essentially this chunk, line 910, you can see is an lapply function. It's filtering over our list. So it's going to apply everything in this function here to each of the Seurat objects in turn. Essentially what we first do is define the empirical cutoff values. So we find the median, for example, of log 10 counts, the median absolute deviation of that same log 10 counts, and then we calculate our lower and upper threshold scores using that logic that I just mentioned earlier. So we do the same thing for the counts features, the counts in the top 20 features, and the percent counts in the mitochondrial genes. You'll notice that there's no lower value for the mitochondrial. We don't want to filter out cells with a low amount of mitochondrial UMIs mapping to mitochondrial genes. Okay, so then after that, we have this chunk here that identifies the cells that we want to exclude. And so the first thing that it does is basically creates indicator variables for whether or not you might exclude the cell based on each of the metrics. So for example, if the log 10 counts is less than the lower threshold, or the log 10 counts is greater than the upper threshold, this returns true, and if not, then false. So that section there creates indicators for each of the metrics. So those are, basically identifying outliers in a single metric. And then this chunk here, or this section here, creates a final exclusion flag. And essentially what it does is there's an if-else statement here where it's actually summing each of the individual exclusion Boolean vectors. Since in R, if you sum a Boolean vector, it actually just returns the integer of the number of true values. So in this case, we're just testing if that sum is greater than or equal to 2. If it is true, then we return true. If not, then it's false. So essentially, this final exclusion flag is defining the cells that we're going to exclude as outliers based on at least two of the metrics that we specified. Okay, so then we go ahead and actually do the filtering itself. So while we sort of defined cells that meet at least two of the conditions as outliers, in practice we actually kind of do the inverse. We're keeping cells that do not meet at least of those two conditions. So to subset, we essentially take our current Seurat object. Again, that's the Seurat object for this iteration of the loop. We pipe it to the subset function, and we're subsetting on the counts. That's the only data that we have at this point. And our condition is simply that excluded is equal to false. So that will keep all of the cells that were not considered outliers. Okay, so then the rest of this is sort of like examining or reporting, I guess, the output from that filtering step. So this kind of seemingly complicated function here is just essentially finding the column names in the original Seurat object that are not among the column names in the filtered object, right? So those are cells that were filtered out, essentially. Dividing it by the total number of cells in the original object or columns and multiplying by 100. And essentially just printing that value as a nice little message that we get. So that'll basically report the percentage of cells that were filtered out in our filtering step. Always good to sort of check to make sure that this type of thing makes sense in terms of the percentages. And then as an additional checks, what we're going to do is actually create two scatter plots. So one is going to be the percentage counts in the top 20 features against the percentage mitochondrial genes. And we simply take the original Seurat object. Actually, sorry, I skipped this here. We're actually going to first create an indicator of whether the cell was filtered. So that indicator is going to live in the original Seurat object. And we're just creating a new column here called filtered. And that's going to be a factor. And essentially, it's identifying the cells that were filtered from the original Seurat object, basically column names that are in the original Seurat object that are not among the filtered object. And since the rows of the metadata are the same order as the columns of the counts matrix, this works. And then we're just going to reorder the levels to make true first. Kind of easier for the plots to do it that way. Okay, so then we just make these two plots. Again, percent counts in top 20 features versus mitochondrial genes. Essentially, we just pipe the metadata data frame for the unfiltered object to ggplot, and we say X and Y, and we're going to color it by whether the cells were filtered or not. That's this factor that we created right here. And the second scatter plot is going to be the log 10 counts versus log 10 features. Very similar looking code here. We're just taking the metadata data frame, piping it to ggplot, and this time we're defining X and Y differently, counts and features, and then we're coloring it again by filtered. We can combine these two plots into a single plot using the cowplot package, which I often use and recommend. It has this function called plot grid, and you just give it a list of any number of plots that you have, in this case just two, and you can say just create a single column, so that'll make the plots kind of stacked. And yeah, so that takes care of the plots. We're saving that as this object plot.list within the function. Again, that only lives within the function and will be erased for each iteration. And then finally, what we're going to do, sort of atypical for this workflow up to this point, we're actually going to return a new list of two things. So the first item in this list is going to be the filtered Seurat object, and the second item in the list is going to be the list of the combined plots that we created in this step here. So you'll notice that actually this whole thing gets saved as this thing called filtered.out. So I'm not overwriting the Seurat list filt in this case. I'm just saving as a new object. You can see as we run this, it prints out the number of cells that were filtered out. And we can take a look at the scatter plots that were created for each of the objects. So if we loop over filtered out in this code chunk here, and we just return the second list item from each of those items in this filtered out list, we'll basically just print out the plot that we created. So it's kind of cool. We can see which of the cells were filtered out, which are red in the plot, and which were kept, which are blue. And you can see that we're filtering out these extreme outliers, whether it's log 10 counts or features, or percent mitochondrial cells with high percent mitochondrial being filtered out here too. Okay, so then to go ahead and actually extract the filtered Seurat object, we can do very similar thing that we just did with the plots, where we loop over this filtered out list, and instead of returning the second object, we just return the first object, which was the filtered Seurat list. And this time we will overwrite Seurat list.filt with it. Okay, so now we have an updated version of our filtered list, and we can go ahead and re-examine the QC metrics like we did before using those violin plots. And we can see that there is a shift in a sense that these plots no longer have such extreme outliers. I would say that the metrics we used and that are recommended by Germain et al. Are somewhat permissive. You can see in the mitochondrial plots, we still have some cells that do have high percent mitochondrial. This plot maybe goes up to like something like 20, 28, or almost 30%. But if we actually go back and look at the original plot for that same sample, some of the cells were all the way up to 80% mitochondrial. So we did filter out, but to some extent, these are permissive values. You could consider doing stricter filtering. Okay. I did want to mention filtering genes by type. So we're not actually going to apply this here. It's really just for illustration. But if you were interested in filtering out genes by gene type, you could do it in this way if you wanted to. So this code here essentially loops over the list of filtered Seurat objects like we did previously. It identifies mitochondrial and ribosomal genes, essentially just making a vector of gene names. And the way that it does that is using this grep function here. We touched on the grep patterns previously. This is essentially looking for all genes that start with MT dash as a gene name, which is the case for all mitochondrial genes. Is looking for those gene names among the row names of the current Seurat object, which is all the genes in that object, and it's returning the match pattern. So those will basically create two lists or character vectors of mitochondrial and ribosomal genes, and then we can actually subset our Seurat object to filter out those genes. So in this case, we're actually using the square bracket notation for subsetting, which is like base R. What we do is in base R subsetting, if you put conditions before the comma, it will filter out rows, and any conditions after the comma will filter out columns. In this case, we want all the columns or all the cells, but we want to filter on the rows where the row names are not among the mitochondrial genes and not among the ribosomal genes that we just identified previously. So that will return a new subset Seurat object, and this message just returns the number of genes that were filtered out as the difference in the row numbers. So you could return this filtered Seurat object. I don't have that activated; it's just commented out. But you can see there's like 108 to 110 genes, depending on which sample you're looking at. Okay. I think that takes care of this QC filtering section. Are there any questions that I can answer at this point on this section?
So I've been doing something similar to so I think I say a hard cutoff for like 15% mitochondrial gene, because I guess we commonly doing it. Yeah, I guess like the rationale here is that even if one cell has the height for one of these QC metrics, everything else is normal, it's probably not a weird cell.
Yeah, so I think you can look at that Germain paper. They do a pretty good job explaining why you might not want to have a global cutoff. Do you have multiple samples in your data set, or is it just like a single like assay?
There's multiple samples.
Okay. Okay. So I think a couple of points are that, you know, the metrics can vary by sample. So I think having a global cutoff for all samples could be I guess, yeah, maybe somewhat oversimplistic in a sense that the distributions of the cells might differ depending on which sample you have. And you actually can see that in like these plots. If you know, look among them, there's definitely differences in the distributions. So I think the strategy is essentially using empirical distributions to define what are outliers based on that sample's distribution. I think is good. It sort of essentially adjusts your filtering sample to sample. And then, yeah, I think the cutoff of 15%, I wouldn't say there's anything wrong with that. I think you might want to play with different cutoff values. And I think Germain, that paper that I mentioned, they do a really, really thorough job of comparing a lot of these different kind of choices that you have in these pre-processing steps. And they sort of come to a good kind of default set that they consider to be pretty good in most cases. And I think they have, I forget how many test data sets they're using, like 6 or something. So I think in a sense of, it's a little bit more vetted and a little bit more customized of a way to do the filtering. Okay, great. I don't see any questions on Zoom, so we can move on. Okay, so this is the last step in this upper section of the pipeline where we have the samples separate. I will warn you, this step is probably the most we'll be going into a lot of depth for this step. So just as a warning here. So, okay, so this step is normalization. And I think we can think of the raw counts for a gene in a cell as being influenced by quite a few factors. There's obviously biological factors that can influence the gene expression, cell type, the cell cycle stage can have an effect on expression. Gene length actually can determine how many reads might be mapped to a gene. If it's longer, then there might be more reads that are mapped to it, or UMIs. And then other biological conditions, right, like tissue type or any number of conditions. Obviously, there's going to be like differences among people, right? People are just different biologically. So if you have different samples, you might have differences. There are also technical factors that we think about. So sequencing effort is sort of like the main one we're concerned with at this step. And droplet size is another technical factor. So sequencing effort, right? Like we didn't do the same amount of sequencing in every cell, and we didn't do the same amount of sequencing across samples, just because there's like a lot of random sampling that occurred during library preparation and during the sequencing phase. And so, but our goal is to compare expression across cells and ideally across samples if we have them. So we really are left with this kind of need to normalize across the cells in terms of sequencing effort. I will sort of interchangeably use the word effort, sequencing effort and sequencing depth. Depth you can think of as just the total number of sequences or UMIs that were observed within a cell. Okay, so that's kind of like our goal and where we're headed. So normalization, I think the Surratt authors kind of like lay out 2 primary goals of having a kind of a quality normalization happen. So the normalized expression should ideally be independent of sequencing depth or effort, and ideally other or possibly other technical factors. So if you are able to account for those, you might be able to sort of remove that dependency. And then the variance of the normalized expression of a gene across cells should be independent of the gene abundance or of sequencing effort or depth. I would kind of characterize normalization in single-cell RNA-seq as sort of following 2 main approaches. There are sort of these cell-specific size factor methods, which were inspired by bulk RNA-seq normalization. DE-seq2 and EDGE-R are sort of like very prominent methods in bulk RNA-seq. There's also this basics, which I've never used personally. And then this kind of alternative method is sort of probabilistically modeling counts to adjust for sequencing depth. SC transform version 2, which I'm just going to call SC transform for the purposes here, is one method, and sanity is another, I believe, Bayesian method. So we're going to be focusing on SC transform, which is from the Seurat authors. It's very well integrated into Seurat, so it's really convenient to use, and it has really good performance. So essentially what the Seurat authors did was find a couple of shortcomings of that first kind of series of methods that I mentioned, the cell level size factors. They found that the cell level size factor normalization is inadequate for a lot of genes. Essentially, they found that the relationship between sequencing depth and expression remained after that type of normalization. So genes with high expression showed strong relationship with sequencing depth. And that normalization also failed to remove the relationship between variance and depth. So we can kind of see from this figure from their paper what they're talking about. What they did on the left has actually been all of the genes into 6 categories by gene expression, by the mean gene expression. So this red category, the darkest red is the highest expression on the right, and the light yellow or orange is the lowest expression group of genes. So then when you look at a graph of the total UMI counts, which is like the sequencing depth for that cell, against the scaled log normalized expression using this cell size factor method, you can see that for lowly expressed gene groups, like 5 and 6, which are these yellow and orange, there is kind of really not much of a signal, not much of a pattern left over after that normalization. But as you move up from, to group 4, group 3, group 2, these are more highly expressed genes across the cells. There remains this pattern between total UMI count and expression of those genes in that group. So essentially, they took this as evidence that these size level factors are kind of inadequate for working with these highly expressed genes, and their method basically aims to overcome that challenge. So their method is called SC transform. It's fairly recent. They first published on it in 2019. Essentially, it uses generalized linear models, and I'll get into that. But essentially, it also combines normalization, variance stabilization, and data correction via regression, if desired. It was actually more recently refined / updated in 2022, based on this paper from 2021, Laus et al., which is the second reference down there. It's also, performance-wise, been found to systematically outperform other methods tested according to that Germain et al. Paper, and it's particularly strong for identifying highly variable genes and rare cell subsets, according to that Laus paper. So those are claims made by people that are not the authors of it. So overall, I think it's a pretty good method, and it's statistically kind of interesting too. So we will walk through the nitty-gritty details here for you all to see. So the first step is to fit negative binomial generalized linear models. Essentially, what this is doing is trying to estimate the gene level proportions using a couple of different pieces of information. So I'm going to walk through this, and I will sort of stop me if there are any sort of questions as I go along. So essentially what we have, if KGC is the count for gene G in cell C, we can model that as being from a negative binomial distribution, which is a distribution that people commonly use for positive integers or counts. We can parameterize that negative binomial with 2 parameters, mu GC and alpha G. So in this case, mu GC is the mean of the distribution, and alpha G is actually like a dispersion parameter. Not exactly the variance, but it is something related to the variance. And so basically the expected value for KGC is that mu GC, the mean. And we're actually going to model the mean as a function of PG and MC. So PG is the proportion of total UMIs for this sample in gene G. So essentially, if you took all cells and all UMI counts and found the proportion that were in gene G, that would be the estimate for PG. Again, so for the expectation of KGC, which is the expected number of counts, we're taking that proportion for gene G and we're multiplying it by MC. MC is defined down here at the bottom. It's essentially the total observed UMI counts in cell C. So it kind of makes intuitive sense. For this current gene in this current cell, we can take the total number of UMIs, total number of counts that we find in that cell, and multiply it by this proportion, essentially, for that gene, where the proportion is estimated across all the cells. And that will essentially, find a good guess for the number of counts for this gene in the cell. Okay, so the way that the authors model this kind of looks a little bit strange. They essentially, so you often have like a link function in these generalized linear models. In this case, we're actually using log base 2 as the link, or exponent of 2. So we're actually calling the expectation for KGC as 2 to the eta, where eta is like a linear predictor term. So that makes basically the log base 2 of mu GC equal to eta, which is equal to the log base 2 of PG plus the log base 2 of MC. Since eta is our linear predictor, it's a little bit easier to think of it as sort of like standard regression coefficients. And we can basically consider this first term of log base 2 PG. The authors consider that like the intercept term. So we can just call it beta naught G for the purposes of this. And then the second term actually doesn't have any unknown parameters, since MC is an observed number of UMI counts in the cell. So that doesn't need to be estimated. That's actually just a value that's fixed and known. So again, eta is this intercept term plus a constant term that we know. So I guess all in all, this whole procedure is going to be estimating 2 parameters per gene, estimating the beta naught G, which is this intercept term, and alpha G, which is this dispersion parameter in the negative binomial distribution. Are there any questions so far? Okay. Okay, so the second step, once all of those models are fit for every gene, we're going to have a parameter, a beta naught parameter for every gene, and a dispersion parameter for every gene. What they do is they do this step called regularization. Essentially what they found is that for lowly expressed genes, and I can show how this figure shows this, on the x-axis is the expression, the average expression for that gene. And on the y-axis is the parameter value for beta naught, in this case on the left. And then the color is actually sort of the standard error for that estimate, right? So low standard error, it's inverse, the uncertainty is inverse with the standard error. So these genes on the left side have a high uncertainty, a high standard error, and on the right side have a low uncertainty, a low standard error. So when they looked at this, they found that the genes that are lowly expressed have high uncertainty, high standard error for the estimates for beta naught and the dispersion parameter. And that kind of makes intuitive sense if you have few non-zero observations, right? That's what you would see if you have a lowly expressed gene. It's just you have high uncertainty in your estimate for that gene. So what they did was essentially shrink their estimates for each of the genes toward this overall mean across all genes. So they fit this lowest smooth regression spline here, which is these pink lines here. So they essentially, it's like a local averaged line. They fit that on this figure here, and then they actually just kind of shrink or nudge the parameters to be equal to that line. So for a specific gene with a specific expression, they will just shrink the parameter for that gene to be the average on this line. And that tends to help these lowly expressed genes have a better estimate for their parameters. They call this sharing information across genes. I guess it is that, but okay. So an optional step at this point is to regress out technical noise, which you can think of as like removing uninteresting sources of variation using the regression. So again, we might have cell cycle, we might have biological sources like cell cycle gene expression, mitochondrial expression, total number of features, and technical sources could be like a batch identity if you wanted to, or other things that you might have as you might think would have technical sources of information. I would say be careful here. Lucan et al. Found that removing some of the biological sources like cell cycle expression or mitochondrial expression can remove helpful biological signals. They point to the fact that, or the example that proliferating cells can be identified by cell cycle genes. So it really kind of depends on your research question, what you're ultimately trying to find. You may or may not want to do this optional step. And then they also point out that given like dependencies among biological processes, removing one signal might actually sort of mask another signal. And then Germain et al. For their part found that it may reduce the effectiveness of normalization. So for example, they saw that when they regressed up mitochondrial gene expression and number of genes per cell, they found that that actually increased the association of the first principal component axis with the total UMI counts, which is not what you would want to see. So I think this is an optional step, and I would probably try with and without to sort of see downstream what effects it has. Okay, so once we have our regularized coefficients from these generalized linear models, we can go ahead and compute Pearson residuals. And so I'll kind of walk through what this Pearson residual looks like here. Essentially, right, we have our log 2 mu CG hat, where the hat is the estimated version of it. That's going to be equal to beta naught G hat tilde, where the tilde version is the regularized version, the one that's shrunk toward that overall mean. And then we have our log base 2 of the total UMI counts for that cell. We can think of mu GC as being 2 to the power of that linear predictor with our estimates plugged in. So for a negative binomial distribution, the variance or sigma hat GC is going to be actually just the mu GC plus mu squared GC over that dispersion parameter alpha hat tilde, where the tilde is regularized, and the square root of that. So once we have our mean and our estimate of the variance, we can actually find our Pearson residual ZGC, which is equal to the observed number of counts, KGC, minus mu GC over sigma. So essentially this is like representing normalized expression values, and you can think of high residual values, or high Pearson residual values as being kind of containing biological information, essentially under the assumption that if we had adjusted for technical variability, then what's left over is biological variability. And again, Laus et al. Found that independently that these were sensitive to biological signals. Okay. Are there questions on any of that?
So Rod also just has a normalized function, normalized data function.
Yeah.
Remember, we had to integrate a bunch of data sets in certain points or something. I think I have used that function to the FET transform. Is that generally, I can't remember the FET transform apparently is really robust or whatever. What is your thoughts though on just maybe the normalized?
Yeah, so I personally have not used very much the regular normalization function. So they do point out, right, that in this first bullet point, the SC transform actually does that. It essentially replaces that normalize and VST. You would previously have run each of those in succession. The SC transform essentially does both of those. I think I would argue that SC transform, just from people kind of looking at it, these papers that I've cited, and I think there are others, find that it's kind of independently a very good way to do it. So I think I would probably just kind of completely replace any workflow that uses those normalized previous versions. Okay. let's see, we can kind of cover how that data correction step gets introduced here. So, right, we have, if we have some additional, covariates, let's call them, that might contain technical sources of variation like batch or mitochondrial gene expression, essentially what happens is we just include them as additional terms in the linear predictor. So if eta previously was the intercept plus the log 2 of the counts, and we now have like a, beta 1, XC1, beta 2, XC2. So those get essentially used in the linear predictor to predict this mu hat GC. And then we have updated versions of the sigma and of ZGC. So in our workflow, we're going to correct for cell cycle scoring. I wanted to show you how Seurat does that. I'm not saying you have to do it this way for all of your workflows, just for illustration purposes. So correcting for cell cycle scoring, essentially there's gene expression that can vary by cell cycle. And so you can regress out the expression levels that are of genes that are associated with those cell cycles. So Tyrush et al. Provided some curated list of genes that they found to be experimentally associated with the cell cycles. And Seurat has the cell cycle scoring method. I think it's probably worth going over what it's doing. So essentially what it does is for a list of input genes that are associated with something that you're interested in, like let's say the S phase for a cell cycle. What you do is you actually first find a set of control genes for each target gene. So you find the average expression for each gene in the target list. You bin all of the genes into 24 categories by average expression across all cells. You identify the target gene's bin according to the expression value of that gene. And then you randomly sample 100 control genes from that same bin. So essentially you're finding like matched pair genes based on expression levels. And then once you have that list, you actually just find the average expression across all of the target genes, let's call it mu T bar, and all of the control genes that you selected, let's call it mu C bar. Then the score for that list is actually just mu T bar minus mu C bar. So it's sort of just like, think of it as the expression of these genes like above and beyond what this list of control genes has observed. Okay, so then when we are running SC transform, essentially what happens is a second negative binomial regression is performed using the scores as covariates. So you can imagine we had previously already estimated beta hat G. We had regularized that beta hat G. Then we actually run a second regression where beta hat G is fixed at the estimate that we found. Again, this log, that should be log 2 of MC, is a fixed parameter. We don't need to estimate that. But then we have these two additional parameters here, beta hat 1. SC is going to be like the cell cycle score for the S phase, and B2 and B2 hat and is associated with MC, where M is like the M1 phase. So then we have new versions of our sigma hat CG based on this new version of mu hat CG. The dispersion parameter would be the same as before. And then we can have updated Pearson residuals. Okay, so whether or not we had done additional regressing out covariates, the Pearson residuals are clipped by the Seurat method. It's a little bit maybe like subjective here, clipping the Pearson residuals. Essentially what they found was that these Pearson residuals are like very, very sensitive to a gene being highly expressed. So much so that they found that if they clipped them, they actually avoided those highly expressed genes sort of like being outliers, like they were almost too strong. And Laus et al. Actually kind of backed this up independently, saying that it does make sense to avoid, basically, there's kind of like some noise because it's so sensitive that you can find in weakly expressed genes that the Pearson residual can be like too high, so to speak. So anyways, what they do is clip the Pearson residual ZCG to something called Z tilde CG, let's say. And it's essentially if ZCG is positive, they'll find the minimum value between ZCG and the square root of N, capital N, where capital N is the total number of cells. And then kind of similar logic where for if ZCG is a negative, you find the maximum of whatever ZCG is or negative square root of N. So yeah, so there's just essentially just clipping the Pearson residuals down to this value of square root of n or negative square root of n. Yeah, I don't have much insight beyond sort of just like these overly expressed, or sorry, weakly expressed genes having large kind of residuals here. Okay, so once we have all of these parameters estimated and the Pearson residual is created, we want to usually find a set of variable features. So the goal is to select features for downstream analyses that contain most of the biological variation. And the rationale is that features that vary strongly among cells after adjusting for technical noise are likely to contain, biological information. So what SC-transform does is actually ranks the genes by the Pearson residuals, essentially effectively identifying biological signal. And then it chooses the top n, and actually you can choose n. The default is 3,000 per sample, but you can vary that. I think 3,000 to 4,000 is a good ballpark to use. I think Germain et al. 2021 recommended 3,000 to 4,000. And typically you start with like something like 10 to 15,000 genes total on average. I will have a note here about multiple samples. So when we run this, and we'll see this in our workflow, right, SC-transform is actually applied separately on each sample. So the kind of outcome of that, right, is that each sample might have a different set of variable genes. So what SC-transform, I guess, yeah, Seurat has a function built in to combine, basically create like an overlapping list of variable genes. Essentially, it ranks the genes by the number of data sets that they're variable in, and it breaks ties using the median of sample-specific ranks, and it finds the top n genes according to those rankings. So, like if if a gene was identified as being highly expressed or variable in 6 out of 6 samples, then it would almost certainly make that final variable features list according to Seurat.
Could you remind me what the variable features specifically contribute to downstream analysis as opposed to non-variable? Like the others, the non-selected genes there, you can factor in all, like clustering or anything else?
Yeah, so I would say it's up to you, right? You can kind of do whatever you want downstream. Seurat, what they do by default is they take these top n genes by the Pearson residuals, let's just say 3,000, and they actually, like for the next step usually is PCA, and we'll cover that. They will use only those 3,000 genes for PCA, and they actually will use the Pearson residuals themselves, not the count data. So under the idea that the Pearson residuals have like this strong biological signal relative to noise contained within them. So they kind of filter on genes and they filter on the signal by using the Pearson residuals downstream. However, I think I do have a warning about this, but right, like if you have clusters that are possibly identified by like weakly expressed genes, that weakly expressed gene or set of weakly expressed genes might not actually make it into the list of top n features. I actually ran into this myself in a project I was working on where I expected this cluster to be a part, and it wasn't until I looked into the fact that, oh, it wasn't actually among these top n genes. So I think it's like, you gotta be particularly careful when you're using just the default values from Seurat. You really have to make sure, you're thinking about your biological question in research. Okay. The other thing I wanted to mention is that SC-transform kind of does this somewhat unique thing. It finds what it calls corrected counts. I guess I would maybe also term them normalized counts. So essentially what you can do is instead of using those Pearson residuals, and again, the Pearson residuals are actually like on the real number line, they can be negative, positive, and they're not integers because they're actually like a scaled residual value, like a difference between the predicted and the observed counts divided by the variance of that negative binomial distribution. So if you wanted to, or I guess they give you the option to essentially reverse the regression and create new predicted, I'm going to call them predicted counts based on that negative binomial, those parameters that you fit. So what you do is fix the parameters in each cell to the median value and then find a predicted count matrix that you would have observed. And what I mean by that is, if you look at the equation for mu hat GC, right, we have this estimated intercept term. Then we also had log base 2 of the observed counts for that cell. What you can do is actually just fix that MC to some value across all cells. So if you picked, let's say, the median total UMI counts of all cells in that sample and fixed MC to that median, call it M naught, then essentially what you get is a predicted mu, call it mu hat tilde or prime, that's predicted that you would have observed if you had observed that median count in that cell, that median depth. So essentially what you're doing is saying, like, what if we had observed all the exact same sequencing depth across all cells? What would our mu hat be in that situation? And then you can come up with a sigma hat prime GC using the mu hat prime GC and the alpha that we estimated. And then once you have those two, you can actually kind of do this neat trick where you convert the Pearson residual back to a count. And they use the floor function here. So what they do is they take the Pearson residual for GC, multiply it by the sigma, which is the variance, and add back the mean. So right, you're sort of like reversing the Pearson residual back to the count. And then this actually won't give you an integer, so they take the floor of that value as kind of like the closest integer value. And they call that, let's call it K prime GC. So they actually put this corrected count matrix in a separate data slot for you if you want to use it downstream. And they also give the option to do this for all genes, not just the variable genes. So you can sort of like have a corrected count matrix that contains all of your genes from the original, which is very useful.
What function does that? That's just part of
I'll go through it. It's like prep SCT find markers is the function. So I'll go through it. Okay. Any questions on that before we dive into the code? Okay. Okay, I did actually, it looks like want to just mention here, just sort of like a schematic view of what SC transform does to the Seurat object. So we start on the left here with our RNA assay. Recall that we have metadata and we have counts and a couple of other things that are not worth mentioning right now. Essentially what we're going to do is once we run SC transform, we're going to create this new SCT assay. It's going to live apart from the original RNA, so we can always go back to the RNA. And in the count slot of the SCT are going to be those corrected counts that I just mentioned. And then in the data slot is actually the log corrected count. So they just take this matrix and log transform it. And then the scale data layer is going to be the Pearson residuals, but only for the variable features or genes. So scale data is not the same dimension as counts and data. Okay. And then here's kind of like what the workflow is going to look like. I kind of want to prep you because it's a little bit messy if you want to do cell cycle scoring. The kind of hiccup is that you want to ideally calculate your cell cycle scores on normalized data, but in order to do that, you need to normalize first. So essentially what we do is we take our raw counts, we normalize with SC transform, we calculate the cell cycle scores using those normalized counts. And then we take those cell cycle scores and normalize, kind of like do the normalization again from the raw counts, but using the cell cycle scores to do normalization and correction for the cell cycle scoring. And we'll just kind of like delete or ignore this initial normalization step or output, I guess. And of course, we have multiple samples in our workflow. We have 6. So we're going to be doing that. Like sample 1 is on the left, sample 2 is on the right. Kind of what I just walked through. We'll do that separately for each sample. And then essentially SC transform is going to combine the normalized and corrected counts for us into a single assay. And then that's where that prep SCT find markers that I mentioned earlier, that's going to create these globally normalized counts and corrected counts, where we essentially fix that median depth to be the median observed across all samples, not just all cells within the sample. Okay, so that was a lot. Yeah, any questions, again, before we dive into the code? Okay, great. So let's hop over to the section here. Okay, so I do have some notes here in the code. I just want to note that variable features n is a parameter that you can set to say how many you want to consider variable. It's going to pick the top n genes according to the Pearson residuals. There's also a setting for return only var genes. And so that returns Pearson residuals only for the variable features. You can actually set that to false if you want, and it'll return the Pearson residuals for all genes if you're interested in having that. And there's also this hidden parameter, which I've only discovered from people talking on GitHub, which is this min cells. So essentially any gene that is not expressed in min cells, number of cells, is actually dropped from the SC transform workflow. So sometimes you might actually see that the corrected count has fewer genes than your original RNA assay does. That can be an explanation for why that is. Okay, so there's another kind of nitty-gritty detail that's sort of annoying. Seurat version 5 is the newest version. It came out fairly recently. It actually works different than previous versions. So before, you would actually, SC transform would work on a list of Seurat objects like we already have. The problem now, though, is that they want all of the samples to be in the same assay, and then they want you to split that assay using the split command that we'll see. And so it sort of gets a little bit messy, but we can walk through each of the steps. So essentially what we're going to be doing is normalizing the list of Seurat objects individually using SC transform without correction for cell cycles, like I mentioned. We'll calculate the cell cycle scores using the normalized counts. We'll combine the Seurat objects using the merge function. This doesn't actually modify any of the data in any way. It just sort of combines the different samples into a single Seurat object. And it creates a single RNA assay, an SCT assay with all the samples combined. We're then going to split the RNA assay in the combined Seurat object. That will create separate layers in the RNA assay for each sample. So it's sort of awkward. We're like combining them in order to then resplit them. But then that's kind of the way that the new SC transform works. It wants them to be split by sample. And then we can go ahead and normalize and regress out cell cycle scores using SC transform. That will normalize and regress out the cell cycle scores. It'll find variable features among all samples, and then it will combine the layers for the SC for each sample into the SCT assay for us. Okay, so let's see what this all looks like. So our first step is to normalize the data without regressing out cell cycle scores. So we're going to use SC transform. Okay, first we're going to loop over, as we always do, loop over our Seurat list, the filtered list in this case. We're going to run SC transform using the current sample for the current loop iteration. We're going to normalize the RNA assay. Those are the raw counts. We're going to create a new assay name called SCT. We're using the V2 flavor, which is the newest SC transform. That's the default, so you don't actually need to specify that, but always good to be verbose. I'm using variable features of 4,000 here. I think it's just a decent number. And we're going to return only variable features. Sorry, yeah, only Pearson residuals for the variable features. And then we're going to say min cells equals 3. So we're going to keep any gene that's expressed in at least 3 cells. Okay, so we'll go ahead and run this, and SC transform should print out what it's doing as it goes. You can see some of these messages here should be familiar. It's fitting a, let's see where it is. Fitting a negative binomial regression, using a specific number of genes. It's correcting the count matrix for those genes, and determining variable features based on those Pearson residuals that we talked about. And that says it places the corrected count matrix in the count slot. Okay, so once this is finished, we can take a look at, or we can go ahead and calculate the cell cycle scores. I do break down the output of ST-transform later in the script, so don't worry. We're not going to look at everything that it does, that it outputs yet. We'll wait for a second here. Okay, so it's finished, and we've corrected the counts for each of the samples in turn individually. So then we can go ahead and calculate the cell cycle scores on those corrected counts. We're going to use the built-in cell cycle marker genes from Tyrosh et al. 2016 that Seurat provides. They were updated in 2019, actually. So you can access those in the Seurat package. There's called CC Genes Updated 2019. There's genes that are associated with the S phase and genes that are associated with the G2 or M phase. So we just create a vector of gene names for those, and then we can go ahead and loop over our filtered list, calculate our cell cycle scores using the cell cycle scoring function, and I described what it did previously. And we just give it the S genes for the S features and the G2M genes for the G2M features, and it will give scores for each of those gene sets for us. It does print out warnings if it can't find some of the genes in the predefined list among the genes in our assay, which is nothing we can do about that, so we can just ignore those. Okay, so then the next step here is we're going to combine the Seurat objects. So we basically, essentially, you can think of it as concatenating those 6 objects into a single Seurat object. I want to stress this is not integration. We're not integrating the data in a sense of adjusting anything. We're simply just combining the assays together. So the way we do that, it's a little bit tricky. We're going to use the merge function from Seurat, which has some unfortunate syntax to it. So I'll kind of go over quickly how we actually do that. So the first step is we're going to name the objects in the Seurat list filt according to their sample name. And that's pretty straightforward. You can just loop over the Seurat list, and you can return that @project name slot from the Seurat object, which we had a long time ago from the first session actually named according to the sample name. So we can just loop over and return that vector using sapply and store those as the names for the filtered object. And if we take a look using names, we can see that the list items in that list are now named according to the sample name. So in R, we can slice a list by name using the single square bracket syntax. So this is an example of just me slicing that Seurat list filt, selecting only the list items that correspond to these 2 sample names. So that'll return two objects that are both of class Seurat, and they each have different numbers of cells in them. So those are just the Seurat objects that are corresponding to those 2 sample names. We can also kind of do a fancier subset, and this seems kind of weird to do it this way, but this is kind of what we need to do for the merging function in Seurat. But you can basically you can subset the names of the Seurat list according to this logic, where you're finding the names in the Seurat list that are not among those names that are for the first object, right? So that's the first sample. So what that looks like, if you run this from the inside out, is actually these are the names of the samples that are not the first sample, right? These are the 5 other samples in the list. So if we actually run this whole thing, it'll return 5 Seurat objects corresponding to all 5 that are not the first sample. Okay, so that seems kind of weird to do, but it's all because of this merge function syntax. So the way that they want you to do it, again, this merge is like a Seurat flavor of merge. They want you to provide an initial Seurat object. So we can just use the first one in the list. We can subset it using the double bracket 1. And then what they want you to provide for Y is actually a list of the remaining Seurat objects that you want to merge in. And again, that's just going to be that same function we did up here, which returns 5 Seurat objects that are the 5 that are not the first, basically. So then we can also add our cell IDs, and that's convenient. We already have those names stored as the names of the Seurat list. So it will actually add those ident. If you recall, there's like that ident variable. It will name those accordingly in the new Seurat object. And then we want to keep the normalized data slot, so we say merge data equals true. So if we go ahead and run this, it should take a little while for it to run because we're combining quite a lot of data. Okay, so we now have a new Seurat object called Seurat.all. It is pretty large and it contains, well, we hope it contains everything. Let's just do a quick check here. We can check the number of dimensions. So this first function here just finds the number of columns for each Seurat object in our original list and returns a vector. So we can just take a look at what that is. Right, it's just the number of cells for each sample. And we can just do a test here. If we add up, if we sum those number of cells by sample, is it the same as the number of columns in the new Seurat object? Yeah, it is. We can also check that all 6 samples are present. We can look at the unique values in the orig ident metadata column, and we can see that all 6 samples are indeed present. We can also see that the cell names or column names now have the original sample IDs appended to them. So, right, the first 5 cells in the new assay have the sample name underscore cell 1 through cell 5, which is great because we can then tell apart the cells later on if we need to. Okay, so here's sort of this newer step, at least newer for me when I was kind of looking into Seurat v5. They use the split function here. So what this is doing is splitting the RNA RNA assay of our new combined Seurat all object. And we're splitting by the original ident, right? So orig ident is right here. It's basically we're splitting by sample. And that will actually split the assay into different layers, and those layers are going to correspond to each of the samples. Okay, looks like Elizabeth has to head out. Thank you, Elizabeth. Okay, so we can check the new structure here. We can look at the layers and see that there's now like count.sample name for each of the layers within the RNA assay. And same is true for data. Okay, we're going to want to make the active assay RNA because we can't remove an assay that is active. Currently, the SCT assay is the active one. So we'll make the RNA assay active, and then we're just going to remove the SCT assay by setting it to null. And then we can check the assays. And again, we're doing this because we initially normalized in order to calculate the cell cycle scores, but we don't want to keep that initial normalization step because we're going to normalize again. Okay, so now we have a new Seurat object with only one assay, the RNA assay, and layers are indeed still split by sample. Okay, so now we can normalize and correct for cell cycles at the same time on our split object. And we'll notice that SC transform actually runs 6 different times, one for each sample. Even though we're not looping ourselves over it, it's actually looping for us. And again, when we run on a split object, SC transform also does the following. It finds variable features that are variable across all samples using this function that you previously had to do manually, select integration features. That function basically does that ranking thing that I mentioned earlier. It finds the variable features, ranks them according to how many samples they were variable within, and then returns like an overall variable features list. And then SC transform also combines all the samples into a single SCT assay, as opposed to, right, currently the RNA assay has the layers split by sample. Okay, so this looks very similar to the previous SC transform that we just ran. We're running on the RNA slot again. We're creating a new SC transform assay called SCT, but this time we actually have this vars to regress parameter set here. We're going to be regressing out the S score and the G2M score for the cell cycles, and everything else is the same as before. So we can see that SC transform, once we look here, you can see it's actually initially running on the count slot for that first sample. So it really is kind of like looping over the samples, even though we don't have a loop specified here in this code. Give it a second to finish up here. Okay, great. So it's finished for me anyways. And you can see at the end, it sets the default assay now to SCT, which is great. Okay, so we can explore the normalized data and see what we have here. We can see that there's gonna be raw and corrected counts. And if we look at the RNA assay, it's still split by sample. These layers are split by sample. But if we look at the SCT assay, we have a single assay. Everything is combined together. We have 19,328 features. And you can see it has this additional thing here, 6 SCT models. What that refers to is those parameters, if you recall from the slides, those were fit by sample. So it has actually kind of 6 model parameters stored. And importantly, it has the median UMI counts across all cells per sample stored as well. Okay, so we can go ahead and look at some of the counts to see the correction in action. So here what I'm doing is actually just looking at the first sample, this SRR 186099983. We can look at just the CD4 gene, and we can look at the first 10 cells. And I'm doing that for both the original assay, the RNA assay, and for the SCT assay. I do have to like subset them differently because one of them is split and one of them is not. But regardless, we can see that the left-hand column is just the first 10 cells, the total UMI counts for CD4 gene, and on the right, for the original assay, the uncorrected, and then on the right is actually the corrected counts. And you can see, indeed, some correction happened. For this cell that had 2 counts, it now has 1. For a cell that had 3 counts, it now has 2 in the corrected assay. So definitely things, SC transform did do some correction. So we use return only var features equals true. So the data slot only contains Pearson residuals for the highly variable genes. Again, that's that list of highly variable genes across all samples. And we can look at some of these values. You can see that they are positive and negative, real values, no longer counts. And this is what PCA and other downstream steps are going to be using. And then we can see variable features. Again, these are going to be used for the downstream. This is sort of that set of biologically highly variable genes that we identified from SC transform. And then finally, we can go ahead and do that overall correction. So again, SC transform was applied separately to each sample. So we have these models. You can see the model formula is always being fit log UMI, and we have different number of cells in each sample, obviously. So this prep SCT find markers is going to generate the new corrected counts. And what it does is it sets that MC term to a fixed value across all cells in all samples. And what they choose for that value is actually the minimum median depth across all samples. So you can imagine finding the median depth for each sample, and then they use the minimum value among those. So if we go ahead and run this, it will recorrect the SCT counts using the minimum medium counts that it reports there, which is 3,399. Okay, so our SCT assay is now corrected for the depth across all cells and all samples, and we should be theoretically ready to compare the actual corrected counts, for genes among cells and among samples, at least correcting for the sequencing depth. So at this point, if we wanted to, we could save our work. I think this code here saves the Seurat object as a RDS file, which is a convenient file for working in R. And this was the original end of session 1, optimistically. Okay. I guess just to note, we don't need to save unless you want to. You don't need to save the file at this point because we're just going to continue. Okay. So that was the end of that. We can start with session 2 here. Why don't we, let's take a 5-minute break though. We've been going for an hour and 20 minutes here. So let's take a 5-minute break. And I do have a timer set for 3:10, which I believe is the kind of like best time to go look at the, yeah, I figured we can take a few minutes to get out there. So yeah, let's take 5 minutes right now, take a break, and we can come back then and start with this second half of the workflow. So this is the second half of our workflow. I think it will be a little bit faster because we have a lot of the everything is kind of all put together in our Seurat object now. So we can start off with principal components analysis here. So PCA, linear dimension reduction. So kind of the problem is that there are a lot of genes, right? In our matrix, the number of dimensions is equal to the number of genes. And so ideally we would try to reduce that for complexity's sake and also just for trying to interpret things, also for computational reasons as well. So we generally first do PCA. Essentially, I'm just going to give an intuition for it. I think Pishu covered it in the lecture that she had more technically, so I'll kind of refer to that. But essentially what this is doing is reprojecting the expression matrix via some linear transformation to a new set of orthogonal coordinates where the new coordinates capture the maximum amount of variation sort of in succession. PC1 captures the most variation that it can, and then PC2 is orthogonal to that, captures the second most amount of variation. I will note it's a linear transformation. Essentially all you can do is scale, rotate, and reflect. Contrast that with like other things that we're going to do downstream, which are non-linear. So we're sort of constrained here in what we can actually do. The benefits are that we obviously reduce the number of dimensions. And another benefit is that the top principal components have increased signal to noise ratio, or they hopefully do. They will kind of contain a lot of the biological signal that we're interested in. I have this sort of contrived example here illustrating the like rotation that can occur when you run PCA. This is some built-in data in R of like these 3 species of flower where we've measured petal width and petal length. If you fit a ordinary least squares regression line through that, you find something like this occurs that attempting to capture the largest amount of variation. And if you do a principal component analysis of that same data, essentially what you find is that PC1 aligns very much with that regression line. So essentially it's like rotated the data space so that this x-axis is essentially like the regression line from the original coordinate space. So just kind of an example of how the data can be rotated under a PC transformation. Of course, in this case, we have 2 input dimensions and we're plotting into output dimensions. So it doesn't really reduce dimensionality, but just as an example. Okay, so that's all I'm going to say about PCA. Let's hop over to the code. Open it up here. I would, for you guys, I would rerun the library code chunk here, line 32 in workshop session 2. sorry, if you want to open workshop session 2 in the notebooks directory first. So they have the library code chunk is important, and then this one below it is also important for running the Leiden algorithm. So go ahead and run that regarding Python, etc. You don't need to run line 72, the code chunk in line 72. That's we already have our Seurat all loaded from last session. Okay, so we can pretty quickly and easily do a principal component analysis in Seurat using this run PCA function. We'll want to specify that we use the normalized assay. And then here I'm keeping the top 30 principal components. That's actually recommended by the Seurat authors for SC transform. They argue that SC transform does a better job of capturing biological data, and so you can keep sort of an increased number of principal component axes downstream because they don't capture like technical noise quite so much. So it does kind of print out genes that are positive and negatively correlated with the new principal component axes. And we can also visualize the loadings. Essentially, these are the covariance values of the original genes or features with the first 2 PC axes. With this visdim loadings function, we can just look at the first 2 PC axes, specify the PCA reduction. And yeah, so these are kind of the top genes that are associated with PC1 and PC2. See like CD74, HLA genes, RPL genes, those are actually ribosomal. So if you have a strong gene of interest here, you might find it in these top genes. We can also plot the first 2 PC axes. So we can plot the cells in the first 2 PC dimensional space using dim plot, reduction equals PCA. It will automatically color the cells by the orig ident, which in our case is the sample it came from. And actually one thing that jumps out on this PC space, well, maybe 2 things. One is that things are not really clustered all that well in this space. I would maybe point to the fact that principal components is somewhat constrained to doing linear transformations. And so it can't necessarily always do a great job of finding these high dimensional clusters. And then the other thing you can notice is that the samples are not, there's sort of things are clustered by sample to some extent. So you can see like SRR 186.10057, the pink one, kind of clusters to the left seemingly of this large cloud here where all of the other samples seem to be kind of on top of each other. And then similarly over in this right-hand side, you can kind of see separation by color. So indicating that there might be some sample level batch effects that are occurring in this PC space. Okay, yeah.
The whole scale data warning and scaling data.
Yeah.
Actually, so I was under the impression that after normalizing, you still have to do an additional scale function data for PCA and for like any visualization. Right?
So the Seurat authors, I think are fairly clear, and I can check this, but they use the Pearson residuals as the scale data because they are essentially scaled. If you recall, like the denominator of that Pearson had the sigma for the gene that sigma had.
Yeah.
So essentially, yeah. So they're essentially like the counts are kind of scaled by the variance for that gene.
And that comes from the SBT.
Yep. Yep. And then I think by default I'm quite sure by default when you say SCT assay for run PCA, it will use the scale data slot by default. Although I think you can actually, it might be a good idea to like actually specify that. I won't do it right now, but yeah, I think you could specify the slot just to be double sure. But yeah, by default, I believe it uses the scale data. Yeah, so actually on this warning, I tried to look into why this was happening for this particular data set. 50 features requested have not been scaled. So what I was able to figure out is that these 50 features are among the top variable features selected by SC transform. And if you remember, we did 4,000 top variable features in that second SCT version. So there should be 4,000 variable features that we're trying to put into PCA, but it's saying that not all of those features have been scaled, which to me means that not all of the variable features are in the scale data slot. And I honestly am like a little bit confused why that is from the V5 Seurat functionality. I think it almost, I kind of suspect that it might be like either a bug or something else happening in terms of like SC transform running on the V5 assay type. So it's something maybe I would look out for, and I am sorry I don't have a kind of a really clear answer. I couldn't find a clear answer when I was looking for it, so just to note that. Okay. All right, so that takes care of principal components. We can go back here and move on to, let me just close this one. And we can move on to the next step, which is sample integration. So sample integration is sort of the common term used for methods that try to remove sample level sources of variation that are not explained by sequencing depth. Things like sample handling or library preparation, maybe samples were collected at different time points. Samples often consist of different people or subjects, and so you might have just individual effects that you need to account for. And you know, this was, I think, listed in like 2020 among the top like open challenges for single cell experiments, right? Like when you do multiple assays across different people and different tissues, you find that they don't align very well in these multidimensional spaces, like in principal components or UMAP. And so people come up with these different methods to integrate samples. Essentially, you're trying to kind of align them in the lower dimensional space, and there's various ways to do that. This figures from Luke and et al. 2022, a great paper on comparing different integration methods. I would recommend looking at it. But their point in this figure, each dot here is a different method for doing sample integration. And on the x-axis is batch correction, their kind of definition of the amount of batch correction that's applied. And then on the y-axis is the amount of bioconservation that's applied. Essentially, one of their main messages is that there's this trade-off. You can't kind of do both, partially because batch correction, there is biological signal that you're essentially removing when you do batch correction. So depending on what your goal is, you might use a different method that might do stronger batch correction or might conserve biological signal more. Personally, I found that it is true for an experiment where I had samples that were put under different treatments. We saw that there was like high differences in gene expression by the treatment. But when we used some of the sample integration techniques, we found that those biological signals were completely removed by some of the integration methods. So definitely really be careful when you're using this. Sample integration kind of looks like this. On the left, we have data sets where some samples came from control treatment and other samples in blue came from this like interferon beta treatment. And when you look at a t-SNE graph and you color it by treatment, you might see that they're extremely non-overlapping. And so if one of your goals is to try to compare or identify cells of a certain type that are common to those two treatments, you would probably be interested in trying to align those samples or to integrate them across those treatments. But again, if you're interested in treatment level effects of gene expression, you may or may not want to be doing this to this extent. So I don't expect you to read all of this. This is from Luke and et al. 2022, that paper I mentioned. They give a really good kind of comparison of these different methods, and they rank them by different kind of tasks that you might want to do. I guess on the left side of this figure, on the left side is like the kind of overall highest ranking, according to them, highest ranking methods. Among those, Scanorama and SCVI are two that are pretty highly ranked. Harmony is an R1, an R-based method that is decently well ranked, and they call it the top method for small or simple tasks. So get to kind of see your options here. Notably, some of the better ones are actually only available in Python. So I kind of just created a pro-con list of these three that I think are three pretty good ones. These are among the top for that paper, and I've personally used these top two. So Harmony is fast, low computational costs. It is integrated into Seurat, which is great, super easy. It does favor integration over biological signals. So like on that trade-off, it's going to, I think, err toward kind of making sure samples are integrated over kind of maintaining this biological signal. Scanorama is also great. It's unfortunately only available through ScanPy, like as far as I can tell, and as far as I've used it. It preserves biological signal quite well. So if you have like a data set where you might have different conditions and you want to integrate across those conditions, I think it's a good example to use. The cons are that it's Python only and that it's computationally quite expensive, like on the order of like a day or so to sometimes integrate samples. So can be pretty challenging. SCVI, I have not personally used, but according to Lucan et al., it preserves biological signal well and it's computationally intensive. So it might be worth giving it a shot. I'm not going to cover it here though. Okay, so we're going to be using Harmony. So I'll cover the Harmony steps. So here's sort of just an overview. We can walk through each of these steps in turn. So what Harmony does, it takes the PC space and it clusters cells across samples. And it does this in a way that it allows multiple cluster assignments for a given cell using this like fuzzy, they call it fuzzy K-means algorithm. And that algorithm also tries to maximize the mixture of samples within clusters. So on the right here, we have a figure, kind of a cartoon. The data sets are indicated by the colors and the cell types are indicated by the shapes. And so, right, let's look at cluster 2. We have 3 different data sets, or you can think of them as samples. And this square cell type, they might not cluster on top of each other perfectly from the different samples. But what Harmony is doing is it's first creating this soft clustering where it's going to attempt to find a overall kind of centroid for those samples within this PC space. And hopefully those will be similar cell types. But I guess notably that the cells can have multiple fuzzy clustering. So like this green square might actually, to some extent, with some probability belong to cluster 2, and with some smaller probability belong to cluster 1. So then the algorithm finds the centroids of both the global clusters and of the cluster sample combinations. So the global cluster centroids are like the center of this fuzzy field here with this probability distribution extending outward. And the cluster sample combinations are these dots that they drew on here. So like cluster 1 for the green sample is the centroid is located there. For cluster 1, the purple sample, the centroid is located there. And of course, those are not going to be usually overlapping with the overall cluster centroid. Okay, so then the kind of magic happens. Harmony tries to compute cluster-specific linear correction factors. So what that is doing, and there's a little bit more detail than what I'm explaining here, so it's maybe worth reading the paper, but it's trying to estimate how much a cell's sample identity contributes to its PC score or position. And the way that it estimates that effect or contribution is with a mixture of experts technique, which is like essentially a set of linear models where each cluster gets its own linear model. And so you can imagine sort of the PC axes for each cell can be modeled with an intercept term, beta naught, and with sample indicator terms. So let's say that ZD is some dimension in the PC space, and I is some cell. So that is going to be modeled as a sum of all of these terms across the clusters. But if we focus just on K, let's say K equals one cluster, then cluster 1 has its own intercept term. And it has these terms for each of the samples, right? So like sample S1 could be this yellow sample here for cluster 2. sample S2 could be the purple sample for cluster 2, etc. And so we're essentially modeling the PC score for that cell according to these indicators and this intercept. And they basically say that the beta naught is, right, so that's going to model this overall centroid location. And then these other terms are going to sort of nudge that apart and indicate the different sample centroids. And beta naught, they call the effect or the contribution of that specific clusters like biology apart from the sample effect. Okay, so what that looks like, this is a figure from one of their tutorials. What that looks like is if you have, for example, these 3 different samples. Sample green is called half, sample purple is called jerket, sample yellow is called T293. So the intercept term is this black vector here coming from the origin. So that's sort of representing the PC score for that cluster, cluster 4 in this case. And then those terms that are indicator terms are each of the colored vectors that are beyond that initial intercept vector. And so those are going to represent sort of where the cluster-specific centroids are located. And so you can imagine actually what we're interested in is nudging the cluster-specific centroids toward that overall cluster-level centroid. And the way we can nudge them, quote unquote, is just by subtracting, for example, for the jerkat cells, we can subtract this vector, this purple vector from that centroid and all of the cells that belong to that sample. And that will just like nudge the cells toward the overall cluster centroid. And so once we apply those correction factors to each of the cells for each of the data sets according to those coefficients that we estimated, we just keep repeating that algorithm. We recluster and find new centroids and try to find new correction factors, and we just keep iterating until we don't see any updating to the clustering, so the clusters are stable, basically. Okay, any questions on harmony before we move on? I guess I would mention that harmony, to some extent, is like apart from some other common methods you'll see. A lot of the other methods are like based on this mutual nearest neighbor concept, which I think there's different iterations of how that works. I am not covering that today, but I think it could be useful if you want to use one of those methods. You might want to look into that mutual nearest neighbor algorithm. Okay. So we can go ahead and use harmony to integrate our samples. So we will use this integrate layers function. This is actually a new Seurat v5 function. They're a new way of doing integration. And we can use it on our Seurat all object, and we will make sure that we're using the SCT slot, the normalization method. And then here we can just specify the method we want to use, in this case, harmony. There are other possibilities that you can put here for method, and it makes it pretty easy to try different integration methods if you want to. And then harmony prefers the PC space. We're going to input PCA for our original reduction, and then we'll call the new reduction harmony. I don't know what to say or what to think about these warnings, harmony matrix being deprecated. I think that's a Seurat problem. I don't fully know why that's happening, but it does seem like it runs fine anyways. Converges after 4 iterations. So maybe something to look into. Okay, so we can take a look at our new harmony modified PCA reduction space. It's now available in the reduction slot. And we can see it's a dimension reduction object with all 30 of the original PC dimensions. So it's done its correction in all 30 dimensions. And we used the FCT assay to do so. We can plot the cells in our harmony adjusted reduced space with dim plot. And we can compare it to the original PC plot. And so before, this is the PC plot, we can see that there's sort of that non-overlapping nature of this SRR 186-10057 sample. In the Harmony version of that, you can see it's now been nudged over toward this big cloud of cells. And then similarly over here on the right, we can see that the different samples are now a little bit more well mixed compared to the original PC space. So indeed, it seems like Harmony is integrating and like nudging our cells toward these overall cluster centroids. Okay. Any questions on sample integration before I move on? I think I can, let's see. Next is clustering. Yeah, I think we can cover this before 3:10. Got 15 minutes. Okay. Yeah. Okay, so cell clustering. So usually the next step, once you have your PC space, or in this case, our adjusted PC space, typically you will start clustering cells at this point. And the way that you do it, at least within Seurat, is you construct a k nearest neighbor graph, and you use Euclidean distance in the Harmony adjusted PC space to define sort of distance on this graph. The Euclidean distance is kind of analogous to like the Pythagorean theorem, except it can be multidimensional. So you essentially just take for cell I and cell J, you can take Harmony, Harmony adjusted PC axis 1 for cell I and subtract it from the location for cell J in that space and find that square distance. Add up that for all of the dimensions that you have, and then find the square root. And so Euclidean distance is going to basically tell us sort of like the distance in this multidimensional space. And we also can provide the number of neighbors that we're interested in terms of like quantifying the neighborhood for each cell. So Seurat defaults to creating a 20 nearest neighbor graph for each cell. So that might look something like this on the right, where you have every dot is a cell, and you can see that the cells are connected to nearest neighbors in expression in this PCA space according to Euclidean distance. You can also create a similarity nearest neighbor network, which, Seurat calls it the SNN graph. You can assign weights to the edges of this graph. The edges are just the connections between the cells as the Jacquard similarity. What this does is it basically reinforces edges in dense clusters, and it penalizes edges across sparse regions. And so the Jacquard similarity is actually just the intersection of neighbors for two cells divided by the total number of neighbors for two cells. So it's like the fraction of neighbors that are shared for any two cells according to the total set of neighbors that they have. So of course, if two cells share all 20 neighbors, they'll have a Jacquard similarity of 1. Okay, so then the hard part is, how do we cluster cells in a way that best captures their natural groupings? So one way of doing this is through the modularity index, also called Q. And this is like a concept that's separate from single cell experiments or anything apart from that. It's a way to basically try to cluster things in space. And so what it does is it measures the fraction of observed within cluster edges compared to the fraction of edges you would see if you just randomly shuffle the cell assignments among clusters. So Q, as defined by Trog et al., who basically wrote the paper on the Leiden algorithm, we can break this down. So EC is the observed number of edges in cluster C. Right, so the observed number of connections of cells within that cluster. And we're subtracting off of that this other term here. The other term is actually supposed to represent the expected number of edges in cluster C under some null model of randomization. And so, right, if we kind of find this distance between the observed number of connections within the cluster and some expectation, if we had just shuffled the cells randomly among all of the clusters, the more connections we have, the more clustering that there would be within that cluster. So K is the number of edges in cluster see if the cells were randomized, and M is actually the total number of edges in the whole network. Gamma is this density threshold for determining a cluster. So if gamma is low, you'll have fewer clusters, and if gamma is high, you'll have more clusters. So the goal is basically to maximize this modularity index. So the problem is that maximizing modularity is basically computationally intractable. It's considered to be like an NP complete problem, I think. And so we have to come up with some approximation methods to try to maximize it approximately. One of those methods is the Leiden algorithm. And I'll cover kind of intuitively what Leiden is doing. It's probably helpful to go and look at Trog et al. 2019 if you're curious for more details. But essentially what it does is it first moves cells among proposed clusters. So actually, sorry, first each cell is its own cluster initially. So there's as many clusters as there are cells. And then the algorithm moves the cells among the proposed clusters and recalculates modularity for each move. It keeps the move if modularity increases. So if we're getting better at clustering, it'll keep that move, and if not, then it'll reject the move. And then it does this refine period between step B and C here, where it tries to kind of subdivide clusters into separate subcategories. So you can see in B, the top cells were all assigned to some red cluster. The bottom left were assigned to blue, and this was green over here. So those are reflected in these large blobs on the panel C. But you can see within this like red cluster here, there's actually red and these new green cells over here. So it's finding like subcommunities within the initial clustering. And then it will actually do an aggregation step where it will consider all the sub categories to be new nodes. So those are now kind of like the nodes on the graph, and it will repeat this method of trying to move the nodes among the full graph and try to optimize modularity that way. So essentially it's doing this iterative algorithm, moving and trying to refine and maximize the modularity. And it will stop once the clusters are stable, once they no longer change from iteration to iteration. Okay. We can hop over to the markdown here. So the way we create a k-nearest neighbor graph with Seurat is we use this find neighbors function. We'll make sure that we use our harmony reduction. You could imagine using the PCA space. And then we're going to save our graphs, our neighbor graphs, as harmony nearest neighbor and harmony similarity nearest neighbor, or sorry, shared nearest neighbor. The SNN is the one with the Jacquard similarity as weights. Our k nearest neighbor, in this case, I just chose 10, and then we're using all 30 harmony dimensions. Okay, so we have our shared nearest neighbor graph. Take a look. Yep, and we have two slots within this graph function, or slot, I mean. And then we can go ahead and cluster the cells using the Leiden algorithm with find clusters. We're going to choose our harmony similarity or shared nearest neighbor graph. That's the one with the Jacquard distance. You can tell it to calculate, to find Leiden clusters with different resolutions. This resolution is that gamma parameter I showed from the slide. If you just specify a vector of values here. And then algorithm, you can use Leiden is the fourth version. There's other algorithms like Louvain that are available through Seurat. In this case, we're going to use Leiden. This does take a little bit to run. Any questions while that's running? So then we can go ahead and take a look at our new clustering identities. They're stored in the metadata, and if you use the graph name Harmony SNN, it will kind of append the clustering column with that. And then if you use multiple resolutions, it'll kind of put it as a suffix for those columns. So we can just take a look and see that, for resolution 0.2, it found this clustering here. For resolution 0.3 and 0.4, you might have found different clustering. Okay, great. So that's Leiden algorithm. And all right, so we got 2 sections left here. I see we have 35 minutes. And I do want to leave time for the survey at the end. I believe it's for the full what is this, the section 3 or whatever it is of the workshop. So we can, I think we should be able to finish by then. So I might move along a little bit faster here for these last sections. Okay, so our next step is going to be UMAP. You could also kind of use TISNE here for this step. Maybe broadly speaking, it's a nonlinear dimension reduction step in the workflow. We'll be using UMAP here because it's a little bit newer. I think there's some debate about whether it's better. I've found debate about that, but we'll just stick to UMAP for now. So UMAP is a graph-based dimension reduction method. The goal of UMAP is to project a high-dimensional shape or surface, i.e. the first n PC axes, into some lower dimensional space. Ideally, we want to preserve global distances in the high-dimensional space when we kind of project down into the lower dimensional space. And essentially, UMAP is doing 2 broad steps. It's generating a high-dimensional graph using what are called simplical complexes. And then it's finding a low dimension approximation for that high dimensional graph. So simplical complexes, a lot of this is beyond my experience, but it comes from topology, the study of graphs. And so a simplex, a 0 simplex is just a point. A 1 simplex is a line. A 2 simplex is a 2-dimensional shape, and a 3 simplex is a 3-dimensional kind of shape. And these are all nodes, and they can be connected within this 1, 2, or 3-dimensional space. So the UMAP authors give this example that we're going to try to track here. And so they start with a sinusoidal curve, which we can imagine is the true shape of what we're trying to uncover, let's say. But in reality, we observe these specific data points that are more or less scattered on the true surface or curve with some error term, right? We don't observe the exact surface as it is. So what they do is essentially for every observation that we have, they extend this fuzzy radius outward from each point. And that radius has basically a probability distribution associated with it. And the distance that the radius is extended is defined locally based on the nearest neighbors for each point. So you can see the radius for this point that's more isolated than a lot of others actually extends further. And I believe that there's some term in terms parameter you can specify for how many neighbors the radius captures on average. The connectedness is probabilistic. So in other words, whether 2 points are neighbors is probabilistic, i.e. fuzzy, and decreases with distance. And then they have this condition that each point has to have at least 1 neighbor with 100% certainty. So this sphere or this circle here is that basically this other neighbor has basically these 2 are connected with a probability of 1 if they're overlapping within the sphere. So you have to have at least 1 neighbor with 100% certainty. So from this fuzzy definition of connectedness, they end up creating a high dimensional graph with edge weights equal to the probabilities that we defined with those fuzzy radii earlier. They say that theory ensures that this captures the properties of the underlying manifold, in this case, the sinusoidal curve that produced the data. I can only take their word for that theory because I do not fully understand it. And I do have this interactive example here, which we can try to pop to. So here's sort of an example of creating this graph from these points. So you can see if you like extend this slider, it extends the radius outward. And I believe the radius is computed as the distance to the nth nearest neighbor. So, you know, the 31% would be like 31% of the distance to the nearest neighbor is what you extend the distance to. And then you can slide the nearest neighbors up or down. So based on this like kind of fuzzy connectedness, you can see that the graph can form. And it looks like these aren't actually based on the fuzziness, is just sort of whether or not they're neighbors or not, the edges form. But in reality, in the real UMAP, the edges actually have weights that correspond to those that fuzzy probability. So you end up with this higher dimensional graph. In this case, the high dimensions is 2 dimensions. That's supposed to basically like approximate or capture a lot of the underlying manifold. In that case, right, we have the sinusoidal graph. You can see that the sinusoidal curve, you can see that the graph does a decent job at approximating that curve, which we don't observe. Okay, so then maybe the harder part is actually creating a low dimensional representation of the graph that best approximates the high dimensional version. So the high dimensional distance is actually transformed into low dimensional similarity with some smooth approximation, which I won't get into, but they have this equation or method they use to kind of transfer the high dimensional distance down into this lower dimensional space with some parameters that are involved in how that happens that I'm going to gloss over. But essentially they define best approximation using this loss function, which kind of looks like cross entropy and is very similar to cross entropy, although it's not quite the same from what I can tell. But anyways, so you can imagine like the high dimensional and low dimensional graphs, they actually share the same nodes, and the weights represent the probability of those nodes being connected. So the graphs can have they have the same anchors or nodes, but they have different probabilities between those nodes. And so essentially you're trying to create a low dimensional graph that best captures the same probabilities of the high dimensional graph in terms of connectedness. And cross entropy is actually one way of essentially comparing 2 sets of probabilities. You can compare probability distributions that way. It's actually, apparently it's the same as the Kullback-Leibler distance, just like to some constant. And it's defined in this way where the weight of the edge in the high dimensional graph is WH and in the low dimensional graph is WL. Okay, I think that's all I'm going to say about UMAP, other than that there is a UMAP Zoo, which is kind of a fun website where you can see how 3-dimensional objects are turned into 2-dimensional objects using UMAP. Kind of a fun thing. Okay, so let's jump over to the code for UMAP. And we can simply use this run UMAP function, makes it very easy, making sure that we're using our harmony space. We can specify how many dimensions we want. In this case, I'm just using all 30. And then we can name the UMAP corresponding to harmony in case we had other integration methods we might have used. Okay. So then the fun part, we can visualize the UMAP, and actually we're going to color by our Leiden clusters that we came up with from the previous step. So we can use the Seurat function dim plot with reduction UMAP underscore harmony and group by the sample ID and then the different Leiden clusters. So we can see originally, here's what in the UMAP space, we can see what the different cells from the different samples look like. And it's really great to see that the samples are overlapping to a great extent. There aren't samples that are kind of, outliers, or they don't cluster apart from each other, which is great. Our Harmony integration worked pretty well. And then the different different clustering created by the different values for resolution for the Leiden algorithm. So 0.2 looks like it does a pretty good job of keeping separate clusters colored separately or identified separately according to the algorithm. So yeah, I think it's probably good practice to choose different resolution parameters and see what makes the most sense. Probably this one might be a little bit too high. You can see that there's some it's calling these cells that are mostly grouped within the same cluster as a different cluster. So, this is like a heuristic. You kind of have to choose the resolution that makes intuitive sense. Okay. I do have an exercise here, but I think in the interest of time, we're going to skip that for now. So let's move on to cluster annotation. Okay. So cluster annotation, I think there are two main approaches that I've seen. The first and probably more common one, or at least originally with single cell data, was manual annotation via gene expression. So this involves basically identifying cluster-specific markers using differential gene expression techniques. You can also examine the expression of known markers if you have a set of known markers for the cells that you're looking at. Some methods to do that would be like the Wilcoxon rank sum test to identify genes that are overexpressed among specific cells. You can also use generalized linear models to do a similar thing. The pros for manual annotation are that they're simpler, more control in how they are done. They're easier to interpret in many ways. The cons are that you often require domain knowledge to examine whether overexpressed genes for a given cluster actually make sense. It's often time consuming to do manual annotation, and there's not always a clear signal or a clear answer, in a sense that sometimes a cluster's top upregulated genes are not clearly sort of unique to any specific cell type. An alternative, which I've only been kind of recently delving into myself, is reference mapping. Essentially, the strategy here is to compare the expression profile of query cells against a reference atlas. So people have created these reference atlases where they've collected a lot of cells from, I think ideally like the same tissue type across a lot of samples. They've integrated them all. There's actually a newer method called Symphony that's kind of meant for creating these atlases. And then you can essentially query your cells against that to compare, to find cells that are most similar in that expression space. So some methods for doing this, Azimuth is like a Seurat-based method in its own package. Symphony, like I mentioned, is another method that can be used, and SC Arches is one that I found. I'm not going to talk about any of those because I don't know that much about them at this point. I do know that some pros for them are that they are automated in a sense that you don't need domain knowledge required, and they kind of just run, it's sort of like point and click. They are data driven in a sense that, you're not making these subjective decisions about what cell types are what. Although I guess you do depend on the annotation for the atlas being correct. Some cons are that it's kind of a black box. It's not always clear exactly, why your cells are clustering with specific cell types. And you do need a suitable reference, which is not always true. And it's challenging for multi-condition data because if you have an experiment and the conditions change in gene expression, then the cells are not going to map to the atlas level cells. Okay, so we'll just cover the manual annotation via gene expression today. One thing you can do is identify cluster markers via differential gene expression. So the goal is to find genes that are upregulated for each cluster compared to all of the other cells in all of the other clusters. So one question is, what data do we use for this? My recommendation, and I think that of the Seurat authors, is to use the normalized counts from SC transform. Again, these are the counts that we would have seen with the same sequence effort across all cells and all samples. I do have a note here that SC transform, the normalized counts are actually not corrected for the other variables that you put into vars to regress. Actually, kind of surprisingly that they're not. They only basically are adjusted for the sampling depth. You can, however, account for batch and other variables in like a differential expression model if you want to. You do not want to use corrected counts from integration tools, according to several sources that I found. The integration tools do not preserve the relative differences in per gene expression, so they're not like trying to maintain this like common gene expression across cells or across samples. And I think the recommendation is instead to rely on a statistical model to account for batches in the differential expression analysis. So I guess I maybe didn't mention this clearly, but some integration tools like Scanorama can actually provide corrected counts after integration. So you would not want to use those. So the goal here is to find genes that are upregulated for each cluster. And we're going to use the corrected counts for SC transform. So again, these are those K-hat GC where we essentially have like taken the Pearson residual and reversed it back to some predicted count that we would have if we had observed some fixed arbitrary sequencing depth in each cell. So using those counts, there are different methods we could use to model those counts as a function of the cluster identities. The Seurat default is actually kind of a safe option to use the Wilcoxon rank sum test. It's going to assess stochastic dominance of expression values of one group over another. It's a non-parametric method. It basically works on the ranks of expression for that gene for the cells in each of the clusters. It does not adjust for batch effects or other covariates. So if you wanted to do that, you'd have to use like a different method that actually explicitly models covariates. But I guess just in summary, what you could do for Wilcoxon rank sum, you define two groups, basically a single cluster versus all other cells as your two groups. For each gene, you'd rank each cell based on the expression level. You'd make all pairwise comparisons between cells in each of the groups and count the number of times that one of the groups wins. Some arbitrary reference group you can call XI and another one called Y. And essentially what the test statistic does is count the number of times that one of the groups wins. And you can compare the U test statistic to a known distribution under a null hypothesis to obtain a p-value. You typically will adjust the p-values using some, usually like Bonferroni, because you have a lot of tests being performed, so you want to adjust for family-wise error rate. Also, a note about p-values, people, including myself, have some problem with like clustering the cells by the expression data and then performing statistical analyses on that same data. Considered kind of double dipping, right? You're using data for like two different things. So the p-values from the differential expression analyses among subpopulations are probably like over optimistic. So I think it's probably good to be conservative, and Bonferroni is a pretty conservative adjustment. Again, I guess I would say that these are typically usually like exploratory analyses, so we're not going to be making like super strong inferences on them, but just as a good precaution. Another thing you can do that I mentioned is examine the expression of known markers. So what you probably would do is plot the expression of your known marker genes on the UMAP space and try to subjectively decide cluster identities based on the expression patterns of those markers. You could also examine the differential expression of those known markers for specific clusters. Okay, so we can go over to the markdown and go to section 5, differential gene expression. So this first section of code first sets the active identities to be the Leiden clusters with 0.2 resolution. That's kind of just the one that I preferred in terms of the Leiden clusters that it found. And then we can use this find all markers function from Seurat. And the test we'll use here is the Wilcoxon rank sum test. The assay is going to be the corrected assay, and we're going to use the corrected counts for that test. They have this option to return only positive, only genes that have a positive log fold change in the target cluster. That's pretty useful for when we want to find genes that are kind of uniquely overexpressed for a given cluster. There's a couple of other tuning parameters here. You can return genes that are only present in at least X percentage of the cells in both groups. So you might want to increase this if you're interested only in common genes across cells, or you might decrease this otherwise. And then there's a threshold value here for log fold change. If you're only interested in genes that are like very strongly overexpressed, you might have the threshold be some higher value. You'll get fewer genes that way from this function. So if we run this, we can see that it's going to do the Wilcoxon rank sum test for every gene, and it's going to do it cluster by cluster, and essentially return to us a very large data frame. We can take, I think I saved that data frame as markers. So we can take our markers data frame, use some dplyr to group by cluster. We can slice max the top gene or X genes, let's say, ordered by average log fold change. And then we can kind of relocate a couple things to make it look nicer. And what we end up with here is a data set of 14 rows where it's the top gene per cluster. So this is allegedly sort of an exploratory list of genes where the average expression for that gene is very strong compared to all other cells in that cluster. We can visualize a heat map, which is pretty useful. This heat map is going to essentially take a random sample of cells from each cluster, and it's going to plot the expression for that gene for that cluster and for all other clusters. Don't need to run it. So it's right here. So you can see, like, for cluster 1, the gene was CD40LG. So every like hash here, or every vertical line in that bin is an individual cell, and the color is the expression, where purple is like 0 counts. Sorry, this is the data slot. So we're using log corrected counts. So you know, almost 0 log corrected counts all the way up to 4. And yeah, so you can see that this gene is across these cells that it's sampled from has higher expression, although some of these other clusters don't have 0 expression. CD8 seems to be overexpressed for cluster 2, etc. And then you can overlay your differential gene expression on top of the UMAP, and you can do that by gene. So essentially, you can use this feature plot function, and you give it a list of features that you're interested in. In this case, it's the top marker genes that we found, and it'll essentially make a UMAP plot for you and color the expression by cell in the UMAP. It looks like, I think it takes the log counts, so it's on the same scale as that previous heat map. So you can see, for example, like this cluster in the lower left has TRBV20, like a lot of cells have that being expressed. So this is sort of a way where you can visually look at these top markers and the cluster that they're expressed in. We can also compare individual clusters if we're interested in that. So you can use instead of find markers, instead of using find all markers, you use find markers. You give it 2 identities. So in this case, cluster 1 and cluster 2. We're going to use the Wilcoxon rank sum again on the C transform counts with the same cutoff values, and it returns a list of, looks like I'm asking for the top 5 by log fold change genes that are different among those 2 clusters. You can see that for cluster 1, the average log fold change is like 4.7 for this gene relative to cluster 2. If we have, if we have a set of known markers, we can, for example, like this list, we can try to visualize their expression on top of a UMAP. So I think this is provided by one of the Strat vignettes, but like you can say IL7R and CCR7 might be indicative of naive CD4 T cells. So we could create a list of a priori top markers, and then we can use feature plot to plot the expression in terms of the scale data on top of the UMAP. And let's see if I can move this here. So that's what this looks like. We can try to look for example, CD8A is expressed in a lot of cells, but seems particularly highly expressed in this cluster over here on the right. FCGR3A seems to be expressed in this small cluster on the upper right here. So, if you have these known markers, you can try to use them to, subjectively identify what cell types are within each cluster. Okay, I think that will cover it for me today. I would be happy to take any questions you guys might have overall. And if you'll please, it looks like we have about 10 minutes left. So if you would please fill out the survey. I think I have it in the Markdown as well as the, whoa, that's really big, as the part 3 survey right here. So this should take you to that.
The QR code.
I made a new QR code and it's apparently giant. So I would actually recommend maybe copying and pasting this.